Caroline.
Thank you very much. The discussion is livening up. I like it. And in fact, perhaps part answer that is implied in the next talk will be given to us by Hugo Vrenken from Free University in Amsterdam, the Netherlands. Hugo?
Try and see if I can get this up. Maybe not. Okay. You can see I'm a physicist. Really good with technical things. Does this need to be... Okay, so that's a pretty hard talk to top. I briefly thought of getting my phone out and showing a video of my nearly 3-year-old son going down a slide or something. But I think I should just stick to my slides. If I can find them. All right, so good morning, everyone, and thanks for the invitation to the organizers. It's really nice to be here and see all this inspiring work. Can you hear me? Okay. So I'm going to talk a little bit about how education and public involvement maybe could help patients get to what that means in the end. So first, just to say that I've done some work with pharmaceutical companies. And I'll talk mainly about multiple sclerosis as an example. That's where most of my work, what most of my work is about. So I don't know exactly what everyone's background is, so I thought I should briefly talk about what multiple sclerosis really is. And I'm in imaging, so of course I start with an image. You may have seen an image like this, where in a patient with multiple sclerosis you can see these focal inflammatory demyelinating white matter lesions. But that's not all there is. There are also diffuse white matter abnormalities. You can see some of that here. I'll talk about that a little bit in a while. And then there are also changes even in regions that on normal imaging appear to be normal, but it turns out they're not. There are also changes going on there. And then it doesn't end at the white matter. There's also gray matter damage, including lesions, and we now have imaging techniques that can show those lesions. So if I can just get this pointer, there are lesions here and here and here in the white in the gray matter, in addition to the white matter lesions that you also see in these images. Also diffuse tissue damage. So this is very briefly to explain. This is just measuring a certain property of the tissue and then looking at how that's distributed in gray matter of different types of MS patients compared to control gray matter. This is all in vivo in patients. And you can see that changes occur throughout the gray matter pretty much. Then the gray matter and also the white matter, in fact, also shrink over time in MS. This is generally called atrophy. So you know this probably from dementia, Alzheimer's disease, but it also happens in MS, albeit at a slightly smaller rate. So this is a famous study showing that it even accelerates. The rate of change of volume accelerates over time in the disease. And we can now measure that much more detailedly. Of course, the Human Brain Project also devotes attention to this kind of tool, measuring cortical thickness. We can apply that also in MS. So we know all these things, but how do we quantify them? So one thing is these lesions that I just showed you. We could ask someone to manually outline them. That's actually what's usually done in clinical trials where treatments for MS are tested. Someone has to manually identify or visually identify and then manually outline all those lesions to make sure that you're getting the right assessment of the lesion volume and see if that's changed under treatment. And of course, work is being done on automating these measurements. And this is an example of just a comparison of in this case, 2 different automated methods with, in green, the manual gold standard as a reference here. You can see that you have some disagreement between the methods and also between the methods and the gold standard. This is just another example. You have all flavors, things popping up with one method that weren't considered a lesion by the expert, and also disagreements in what the size of a lesion is, or lesions that are missed by methods. And we're doing studies to quantify these things. As we move to more detailed imaging, we go to 3D imaging with high spatial resolution, or what we in clinical imaging call high resolution, which is 1 millimeter cubed resolution, which is very good because we can see very detailed things, but then the manual outlining of the lesions really becomes impossible because you have far too many slices and it's far too much work. So we're working on automating that segmentation, and you can see here a study that we did where we developed such a method. This is the reference outlined by the expert, and this is what the method tells us the lesions are. So the agreement seems to be pretty good. So is that problem now solved? Well, to answer that, I just want to show this figure that my PhD student Martijn Steenwijk generated in the course of making this automated method, and to explain it, since this is about education today. I thought I should do a little bit of education as well. So this is just to explain the measure that was plotted in the previous graph. Dice's coefficient, which it's very funny because it's used to model, to calculate agreement between things that are in voxels, so 3D pixels, in images. It's a very popular measure for quantifying the performance of methods for outlining structures. So people tend to think, it has something to do with dice that you throw in a game, but it's just actually someone named Dice who developed this coefficient. And it's just a way of quantifying how large the area of overlap is compared to the total area of two sets that you're comparing. So for an ideal overlap, the value would be 1. If there's no overlap, it's 0. And this is very popular because it tends to get high very quickly. So you can always report 0.95, even if there's actually a lot of things that are wrong. So that's the number that's given here. So this is 0 at the bottom and 1 at the top. So even for the best ones, the best method that we got in this evaluation. So different methods of handling the data and adding prior information and varying a parameter, even the best ones give us something along 0.8 or something like that. So there's still a lot of improvement to be made, especially when you're thinking of individual patients whose treatment decisions would depend on such measurements. So the automated methods are still not perfect, and experts remain necessary. Then the diffuse abnormalities that I talked about, we did some studies on quantifying what's going on there. So we did post-mortem work. So this is a translational study. It's really composed of two parts. We had a post-mortem part where we measured in brain slices that were fixed. We measured several things. I'm not going to go into the details. And then we quantified in the tissue staining several properties and found that in those diffuse areas, even though you think on the MRI they look a little bit fuzzy. So this area here it looks a little bit fuzzy. It may be, just a little bit of water that would also give you an increased signal like that. But in MS, actually, this contains axonal damage, gliosis, demyelination, all of these things. So it's actually quite severe. And then we looked whether we could see that also in vivo. So we investigated those areas indicated here with the contours, and found that actually these graphs are almost too good to be true. In two different types of progressive MS. Comparing normal appearing white matter, this diffusely abnormal white matter, and then the focal lesions. It's really a category in between those. Doesn't mean that it's a precursor for lesions, but it's really damaged tissue, as you can see here clearly from all these different indices that we measured. So I already mentioned that brain atrophy also occurs in MS. In view of time, I will go over this very quickly. One of the things you can measure is enlargement of the ventricles. We developed a method for that and looked in MS whether that predicted disease progression on the typical clinical scale for MS, and found that indeed if you measure right after diagnosis during the first 2 years the expansion of the ventricular system, which is sort of the result of the brain atrophy going on. If the brain shrinks, then the CSF spaces will expand. And we found that this was a predictor for clinical progression 5 years later. And we repeated that in a large multi-center study with nearly 300 MS patients with a longer 10-year follow-up, and again found that expansion of the ventricles and change of lesion volumes were predictive of late disability progression. So to measure atrophy in an image, you need to most packages first need to identify, what's the brain roughly, or the intracranial cavity, and what's non-brain. So this is a tool called BET that many people use to extract the intracranial cavity, and you can see here the typical performance of BET, default BET on an image of an MS patient. And BET tends to work very well on healthy control images, but when you have diseased brains with atrophy, there tend to be errors. And as you can see here, so part of the eyes and the orbital fat is included, and part of the cerebellum is excluded. And if you want to measure what the brain volume is, that's probably not a good idea to start by cutting off part of the brain, and also including regions that are definitely not brain. So we did a study to quantify that performance, and this is just showing you in two different images of two different patients the output of various different settings of this software. And Veronica Popescu very elegantly analyzed all this very systematically with different kinds of pre-processing for the images, removing the neck, and also performing inhomogeneity correction, and then looked at how the performance varied. And just to point to what needs to be improved in the tools that we use, this red rectangle shows you the performance of the default version of this software, so the default settings in 3D images of MS patients in 16 different ways of generating the MRI images, so 16 different acquisition protocols. And this is again this overlap measure that I explained before. That's why I took the time to explain it. So this ranges here from 0.5 to 1. And you see that it goes down to very low values, just for finding the brain. So we have a big problem in using that. But the good news is that if you do some standard pre-processing, you can actually improve this very much, and it works very well across all these different protocols. So there is a generic solution. And then when you look at the brain volume that you measure from that, this is the gold standard volume. This is the default, very bad compared to that very large underestimation of brain volume. And then even across all the ones that were very close to it, so had very good performance in terms of overlap, there's only one that's that is not significantly different from the gold standard values that you get. So thinking about individual patients, we're still, there's a lot to be done. Finally, to close off with the brain atrophy, and in this case the hippocampus, in Alzheimer's disease and mild cognitive impairment, there are several tools now that automatically identify the hippocampus, and they do a reasonable job in healthy controls. But again, when you go to diseased populations, the information on what a diseased brain looks like is usually not captured in the models that underlie these methods. So you run into problems, and we quantified how reproducible the measurement of hippocampal shrinkage over a 1-year period is with different methods, including manual experts and two very common automated methods, FreeSurfer and FIRST. And this is just to show, there are some minor errors that we thought were acceptable, but there are also much larger errors that we had to exclude. So some of these automated methods in some images will give you a number, but it will be based on the wrong region, or it will be a gross over or underestimation of the real hippocampus. So that's another thing about automation that you have to be careful about. So what we did in this study was just to compare using two scans at each time point. So we have a baseline scan and a month 12 scan. This is the ADNI1 data. We have two scans at each time point, and just using two pairs of scans we can assess the reproducibility of volume change measurement. So we have a manual expert outlining this. Hippocampus at all 4 scans. We have 2 automated methods doing the same. So the 2 scans were acquired immediately after each other. So I want to ask you what you think. We tend to think that automated methods may be more reproducible. That's why we did the study, to quantify that. So this is the 3 different methods. So which one do you think is the manual measurement? So this is the difference between the 2 measurements, and this is the mean. So it's just a standard Bland-Altman plot. 2 of these are automated. One of them is the manual. What do you think? Which one is manual? So the idea generally is, if you have a manual expert, in the morning he might measure something different from in the afternoon when he's tired, or you ask someone else, you get a very different result. So this is thought to introduce a lot of variability. But I think the plots already show that if you have an image acquired even in the same session, seconds later, and you feed it into the same algorithm, you get a very similar error compared to a manual expert. So the middle was the manual. All right. So to round off, we have a lot of, I think, common challenges and maybe common solutions. So for the lesions that I showed, of course, the question that everyone in MS asked me when I showed that slide is, you have this one gold standard. What if you ask 10 other experts to outline the same lesions? So you probably get different answers. And we want to do that, but it's a lot of work. And how do you organize that? So one thing that you can do is share the workload if you have multiple relators, but you can also increase the confidence. For example, here in this segmentation, if you have a lot of people saying this is a lesion and this is where the boundary is, then you can build probably automated methods also that are more reliable because they're not just based on the interpretation of one expert, but on many. And to do such things, you might also want to involve the general public. So finally, where does this all lead? Thinking as a scientist, our imaging methods for research, I see that they continue to be improved. We're doing great things. We're really understanding disease mechanisms in the examples in MS that I showed. But it's become a bit of a personal frustration for me that in clinical practice, none of this is making any impact at all at the moment. The only thing that's known with imaging in MS is that the radiologist counts the lesions for diagnosis, and that's it. So we want to try and change that, and they're used a little bit in treatment trials, but not so much. So we want to try and change that with a project that we got a group of academic centers and companies together in a project called Mix for MS. It's an application that we're hoping to to receive good word on soon. So apologies to any reviewers who may be here. So I'm not trying to influence you, but just asked to show, what I think we could do. So this is really about education and research together, bringing not only new experts, but also validated imaging biomarkers for multiple sclerosis. So it'll be 15 people working on different aspects of MS pathology throughout the central nervous system that you can capture with clinical MR techniques, and an education program that's based on how we might go from some original ideas to a validated biomarker involving all kinds of things like regulatory affairs and etc. So with that, I'd like to thank the people of my group and everyone here. Thank you for your attention.
Thank you very much, Hugo. Quick question. Yes?
So the general idea of crowdsourcing seems lovely. How do you deal with privacy issues? How do you intend to deal with privacy issues?
Yeah, it's good that you bring that up. Okay, it's good that you bring that up. I'm involved in the Netherlands in an initiative to work towards legislation and advice on how to handle that, specifically in imaging, where this is maybe comparable to genetics, but definitely imaging is also an area where people are worried about this thing, and I think that's correct. It's good to be worried about that. And right now, I think that's holding a lot of people back to really share their data or collaborate in an open fashion, because they're not sure that they're not doing something that's not allowed. So we're trying to create some clarity on what can be done. And definitely things like removing the face from image data where it might be recognized is something that tends to be done, and that people would prefer. But the problem is, again, with the image analysis tools that are now out there, they expect that part of the image actually to contain data, and if you remove it, they might fail dramatically. So you have to either adapt the tools or change the image somehow so that there's still data there, but it's not actually the face that a person has. So things like that. Yeah.